You want a better zoom on this guy?
No, that's enough. Thank you.
Roger. There's another one of your firecracker corals there. Or is that true?
Oh, yes, it is. Yeah.
Sorry. Bumpy
There's
here.
one of them.
So Steve, for a guy like me or viewers who are watching that want a basic marine bio guide for deep sea or really anywhere, is there
Video push in, please.
any kind of course or materials that
That's good.
kind of is succinct but also pretty, basic?
You know,
Deep
iris
sea
down a
bio
bit.
you're talking about?
Yeah, I suppose so. I mean,
Yeah.
everything, I never took a marine biology class or anything. It's all on the job.
Yeah.
So I don't know if there's any kind of resource to hone that in.
There is a resource, and yeah,
Okay, come wide video.
Thank you. Yeah, that zoom is great. Yeah, so there is a guide for certain regions.
Runaway?
Mostly in the
Runaway.
Pacific.
Running over you.
Yeah.
That is run by NOAA's Office of Ocean Exploration and Research. And if you Google benthic Deepwater Animal Identification Guide, you will find it. And in fact, it has everything listed by taxonomic groups, and then it has representative photos of almost every single morphotype or morphospecies of animals we've encountered. Everything from fishes to sponges to whatever, and as well as it tells if we can't identify it to a species level too. Most of the stuff actually isn't identified to the species. So
Awesome, thanks Steve.
guides.
Benthic Deepwater Animal Identification Guide.
Yep. Yep.
I wrote it down.
And the imagery is
I can
pretty good
study
quality.
up. Yeah, it's like mainly just the basic classes and then getting a little bit better after that.
That's right.
That's what I'm after.
If you zoom in one click on the nav screen, Rennie, does it get 20-meter divisions?
On this one.
Yeah,
Two
yeah,
clicks,
that's better.
there you go.
Perfect, thanks.
Yeah, we were zoomed out because the ship was so far away before, but now we're not.
Yeah, the one thing you probably won't find for a lot of these deep sea animals are common names. Mostly because it's not a common occurrence to encounter them, and so they haven't necessarily been given common names. You know, for example, bubblegum corals are well known, so they've been given the name bubblegum corals because of the way they look. But some of the other groups, like primnoids, don't really have. There's in the lower left. There's that broad fan.
The pink one?
Yeah, now it's at 6 o'clock. So it's just to the left of the yellow stick.
Yeah. Okay,
Yeah.
I see what you're
It's
talking
got
about.
a bunch of red ophiuroids, brittle stars on there. It looks a little bit different.
Sure.
I'd like to take captures of that.
Sure thing, Steve.
Now this is the kind of deep water community that is characteristic of this depth. The other sites where we're diving in this depth range, we didn't see any of this.
All right, video, go ahead and do a half zoom there.
This is more of what I'm familiar with for this depth
good.
range. Oh, what do we got there?
Sorry. Go ahead and push in a bit more there, video. It's good.
Okay.
What you're saying, Steve, is it doesn't get much better in this?
It does get better than this.
It does?
Push in there a bit more there, video.
So
It's good.
thanks for that zoom. That's sufficient. This is not the original tenant of this coral colony.
Oh, yeah?
It's a zoanthid. Yeah, so zoanthids are usually some sort
Coming
of
up.
parasitic organisms that takes over a coral skeleton.
Man, they don't do any of the hard work.
No, they don't. Yeah. So they come in there and they usually find a spot opportunistically towards the base of the colony and then grow up and then, you know, essentially consuming over time the architecture of the colony. Difficult to say what that colony was, though. The branching pattern was kind of distinct. It might have been a Pleurogorgia, one of the dominant species right here.
What's that?
So is this waypoint 220801, that intermediary?
Yes, I'll
Oh.
zoom out a couple clicks and I'll
Mhm.
show you. My thought is for this slope upcoming is to track a line at 0.2 knots. That way we have some opportunity because it'll be a little bit more of a sheer slope.
Yep.
We'll have
That's
some time to sample if we need without having to jostle around too much.
Yeah. That's that sounds good to me.
Okay.
Is
Yeah.
it a long? It's a long slope, is it?
Well, it's like 300 meters, is that?
Let's see. From where we are to the top is going to be 580 meters.
Oh, wow.
I'll try to pick a bearing that's kind of perpendicular to the slope. So we're heading right up it.
Sweet.
And
Sweet.
yeah.
Yeah.
We'll try out the 0.2. If it's painful, then we can always increase it. So starting to get more life up here, huh?
Wow. Yeah. No, this is what I'm talking about.
This is what I'm
Yeah, this feels good.
I
That's what I'm talking about.
This is
Yeah.
not even crazy yet, but you know, compared to what we're used to, this is pretty a significant improvement. And
Are you ready to kind of start that bearing 330 move?
Yeah. Yeah, that sounds pretty good.
Does that sound all right, Steve?
are we stepping, or are we just?
We're
They'll be
we're
tracking the
stopped
line.
right now.
Yeah. No, but are we gonna step up towards waypoint 4, or are we gonna?
I was gonna track a line at 0.2 knots.
Okay.
What do you think?
Can we do a few steps, and then
Yeah.
if it seems to be kind of the same, we'll track that line.
Yeah, let's do that until maybe till we get to the base here.
Yeah, because there's a bunch of new stuff here that I want to potentially do some zooms on.
All right, let's go. I'll start to do some steps at 330. Bridge now.
330, Roger.
330. You want me to bring my head
Could
around?
we have a step
Yeah.
30 meters,
Coming around.
bearing 330. Thank you.
Doesn't look like that's uphill.
Yeah, actually, we're going to be pretty flat here for a little bit. On the other nav screen, you can see we got a bit of flat, and then we'll hit, hopefully, in about 120 meters, we'll hit a slope.
The way we were going was that.
I think that was along a ridge strike there, but I think they want to go up to the top of the cone.
You can see that is still going up the way we were going, but we're kind of changing. That way is upslope, but we're going to change direction to go up the big slope.
I see, I see.
We'll be running a little bit parallel to these general lines for a little bit, and then maybe 100 or so meters, and then get to the big base. Of course, right where we're at, the 30 meter, the step that we're doing is going to be sand for a moment.
That's all right. Kind of a cool sandy scape, though.
It is, yeah.
Goes downhill nicely. And
Oh, look, there's a little rock on your right. You can strafe.
Yeah, let's have a look.
Have a look. Little oasis in the desert.
It's pretty cool shape.
Yeah.
Slope ski down that thing.
Yeah, that's what I was thinking. It'd be kind of a cool ski ledge there.
Little lonely corals there. See, they're living on their own island.
Oh. Yeah, come down a little bit there.
Yeah, just thinking the same. Sorry.
No, you're good. Sorry.
Do a pirouette, Jess.
Yeah, see the underside of this rock? It's like a little bit of slack.
You can
On
see
the
how
fly pirouette.
that current is feeding these coral.
Hey, do you think we can look at that largest fan on the far side, even if it's from the distance?
Pirouette.
Yeah, we'll get over there.
Okay. That's pretty distinct. It's not Pleurogorgia.
What's that yellow thing on it?
I don't know.
It's a crinoid, I think, feather star.
I'll come right down and give you some leash.
Yeah, thank you.
Nice little ledge.
The whole forest around there.
It's a cool Argus shot of this rock we're looking at. The little ripples caused by the current around it, how it changes.
Yeah, that's the one I'm interested in.
A lot of life on this side.
All right, video, go ahead and push in
The
there a
right
bit.
half of the colony
Okay.
looks different from the left half. Wonder what's going on there. Zoanthids.
Zoanthids.
Dun dun
Nice,
dun.
thank you.
Zoanthids. Ah.
Yeah, so what we got here is a primnoid octocoral, half covered with zoanthids on the right hand side. Funny enough, they're actually the same color as the colony. But this one has a light rate branching pattern. We get tight on the polyps? Yeah, thank you.
Nice.
So
Nice, Wayne.
Thank you.
we haven't seen many of these types of primnoids, but from how I'm looking at it, there's a shrimp too. One of the ways we can tell different genera, generally, not specifically, there are always outliers to the rule, is the direction that the polyps are retracting or pointed towards when they do retract. And these look like it's pointed upwards when they retract. They kind of bend against the axis at an angle. This would indicate that it's in the genus Calyptrophora.
Do you want to see anything else on this?
Yeah,
part of
the
rock?
pink puffball there.
Yeah.
Dense,
I knew
dense
you were going
polyp
to go
ball.
to that pom-pom. It's just too irresistible.
I think the boat stopped, so you got all the time you want.
Yeah.
Yeah, it should be.
Oh, yeah, okay. If the boat is stopped, how would
Yes,
you
we are.
feel about sampling one or two things here?
Of course. You want
Okay.
to sample? Come on. Yeah.
All right.
Running out of sample space, huh?
Slurp.
All those corals.
Let's test that.
Ready?
Let's test that hypothesis. So I like this pink puffball. I want that pink puffball.
You want
And-
the thing right
Should I
on
push
the-
in on it?
Yeah, please.
Yeah,
Okay.
go ahead and push in a bit. And I'll get a little bit closer as you push in.
You're not Emily.
Yeah, I want that.
Go ahead and push in a little bit there, Lane.
Okay.
That gives you enough light.
Why does it interest you, Steve?
So this is in the genus Chrysogorgia. It's in a group that is it needs more samples to better describe the diversity in this group. If it does belong to the group that I'm thinking of, it's fairly distinct, dense polyps, and I want to confirm the ID on this one because I've seen something very much like it in a different part of the equatorial Pacific, but never got a good identification on it because we never collected a physical specimen.
Okay, so you want to grab this one?
Yes, please. And
How
this
much of it do you want to grab?
I'm spinning around a bit.
Sure
to get
thing.
some better shot.
This is I wouldn't say it's gonna sink like a lead, brick, but it's
Zeppelin.
probably not as floaty as the last two.
Huh, all right. I'm going to see if I can drive into the slope here. See how this goes.
I'm gonna light up the manipulator.
Oh, jeez.
Not too far off the bottom. Might be able to sit. It might not be too high.
I am 1.5 meters up.
You can spin around and land on the rock and reach down, maybe.
Yeah, or so I can drive forward.
Or you can perch and I can nab it.
There. Here, anyone do me a favor?
Yeah.
You want to press that stick lock for me?
Ready?
Yeah, go ahead.
Stick lock.
Thanks.
Craft is ready.
Cool.
Do it, do it.
Do it, do it, do it.
How's that cookie, Emily?
Oh, cookies. You didn't bring any up for us?
Yeah. Party foul.
Bye. It's in my my stash in my room.
Ah, I see.
Although, I did smell cookies on the way up.
It's good. We'll have to do a cookie
So-
run at 3 p.m.
I think our 3 o'clock snack is definitely
Steve,
cookies.
are we going to grab this or slurp it?
We're gonna grab it, and you can
Okay.
take the whole thing. In fact,
Awesome.
yeah.
Did you want to zoom on it before we steal it?
Yes, please.
All right, I'll come out a little bit. Okay. All right, video, go ahead and push in on that coral. Hold that. Bump up. Okay, go ahead and push in a bit more there. That's good.
There's a couple of associate organisms in there too. Shrimp, it looks like. Or maybe squat lobster, I think they're shrimp.
Thanks for the zoom.
All right, come wide, please. That's good.
You can try a little zoom on Argus too, if you want.
Yeah.
Yeah, didn't think about, should have thought about this a little bit better.
Oh, come back, come back.
I'm gonna take out this creep in my wrist here real quick.
Yeah, this one does not have a firm ID yet, so this is a great opportunity for this collection.
You want to zoom in on him a little? If you're bored. That's good. Of course it bounces now.
I'm just gonna calibrate out this rotation in my wrist.
So can I see the sample binder? Just to acquaint myself with
that
what
looks
is
good. There you go.
full and what's available. Yeah.
Sticky buttons.
Yeah, I would go
Okay,
A or
that looks
B.
good.
Yeah.
Okay, thanks for the pause, guys.
So Jess has managed to park Hercules. She's not landed, she's pushed in on the porch there. She's literally balanced on a marble.
Video, you want to push in a bit there, please?
If you grab and twist, it should snap off. It's not a fibrous colony.
Okay. Just scraping against a rock, though.
Mm-hmm.
Keep getting dripped on.
All right. Actually, never mind, sorry, I'll give you guys a picture for a sec.
Just a note for the data. It does look like the shrimp is inside there.
Got it.
Yeah.
Video, you want to push in on that a bit there, please? That's good.
Excellent.
It looks like this might be kind of like a clump of hairs, you know what I'm saying? Oh, that's not good. You want to come a little wide there? So where do you guys want to put this in? They're going to be loose.
Okay, forward bio A or B, or sorry, starboard bio A or B, please.
Okay. I might land this so that we don't kick out a bunch of the corals that are in there. Okay, so video come wide.
You just want to put your footprint in that sand.
I do. I want to make a mark.
Herc was here. The little kid in me so wants you to jump on that sand pile.
All right. You want to be ready on the craft valve in case it keeps on jostling up by itself.
Yeah.
So.
If it starts beating itself to death, just I'll reach over and bash the blue button.
When we get the loss in comms, it actually starts to come in towards the eye, so we have to kill the valve.
Roger. It doesn't respond to the blue LED button?
No, then we had to restart this console here. It's not a good day when that happens. All right. Let's try this guy out here.
I see you have that page up, so I can do the boxes if you want, or are you gonna do the boxes and I'll stand by on the valve? You got it there. I can.
Yeah, if you want to do the.
Yeah, I'm ready.
I'm going to open those boxes slowly too, just in case.
Opening box slowly. I'll wait till Jess gets around there. Ready for salvo starboard?
Yeah, let's do it.
Salvo starboard.
All right, I'm ready when you are.
Ready?
Yeah.
There's
You're
a bump.
I think it's, yeah, that one we just put in there is going to stay there. That's good.
Looks like it.
All right.
Where are you going? One of the back ones?
They want A or B, I think, was the consensus.
A or B, because they have rocks in the bottom, which means that we won't have to put anything on top of them.
Roger.
Copy that.
I'm gonna leave at least one of the big compartments open in case there's just big rocks.
In case a geologist shows up.
I don't know how to spell that. So why? Chrysogorgia? I don't know.
Nice parallels. Rotate.
Sinker.
Awesome.
Perfect.
Awesome.
Can I have a sample ID?
047.
047. And you said this is a Chrysogorgia?
Mhm.
And I don't know how to spell that, but I guessed.
Yeah, you spelled it right there.
Oh,
Nice.
sorry about that, guys.
My Latin is just...
Debris. Closing.
Roger.
Nice collection.
Go, go, gadget. Salvo. Forward. Salvo normal.
I love this shot right now in Argus. Cool.
That's sweet, yeah.
Mm-hmm.
Yeah.
I really want to
So
right in the sand.
I think Steve has another victim in mind here.
Yeah?
Steve, what's that little
Were you
thing
reading my mind there?
right
or-
in front of Hercules, that little worm-looking thing that's wiggling around?
Do you want to look at that?
Wiggling around.
So you might as well leave that.
It's,
Yeah.
on, it's on the
Hot-ish.
seafloor.
Yeah.
There's a star there. You see
It's black. It's in the sand.
black in this.
Wriggling.
Oh, it's wiggling around.
Yeah.
It's basically in the
Right
lasers there.
Yeah.
Yeah, we can take a look.
I want
Sure.
to know.
All right, video push in,
Maybe a
please.
small fish.
I'm going to come out my pen and tilt. Is that as far in as you got?
What is that?
I'm going to do
It's
one
a
bump up.
worm.
Is that a like a baby
Hagfish.
hagfish?
Ugh, slimy things.
No, well, yeah, what is that?
What is that
That's a
thing?
cutie.
It's so tiny and cute.
He's rolling around.
I think it's had a lot of
Laughing.
catnip.
No, okay, so
All right.
this is not a fish.
Yeah.
Oh boy, going back to my invert bio days.
Google search
I'm watching this thing
green
just-
slimy.
It's not a
Evil thing.
polychaete. It's a it's either a peanut worm or it's a priapulid. I'm not sure. But it's usually an infaunal worm. They're not found on the sediment in either case.
Yeah.
So it's kind of weird that
I've
it
never seen one before.
is there. Yeah.
Maybe we dislodged him from the rock.
Maybe. Yeah.
Seems to
He
be.
seems kind of confused.
Seems to be looking for a hole.
Yeah.
Oh, sorry, overshot him.
Yeah, I'm gonna go with priapulid here. But very nice, yeah. I'm not gonna waste a sample jar on that critter.
Roger. So go back to our sampling objective.
Yeah, so I was thinking about this big fan up on the rock to the right, the one that we zoomed in on the way in.
But why is there sand as opposed to sediment? Or does it just look like sand?
Land in it and see if it goes poof.
You know, we did slurp a bunch of sediment with one of the collections, so we can probably find out.
This heavy sediment, the light stuff blows away.
So if you can get me a better zoom on that, we won't need to sample.
Sure.
And then
It's a challenge.
yeah, we'll go with that. Because I don't want to linger too long.
Are you mostly
How about
concerned with the polyps, or?
Tight on the polyps, as tight and as in focus as you can get me.
After
Okay.
touching the camera, it's too close.
How about I perch myself here and then we can zoom for days if you want to.
Sounds like a plan.
Day 43, still looking at
Still
it.
zooming.
Still zooming in on the coral.
Everyone's waiting on the dock in Samoa.
Yeah.
Hey, where'd the ship go?
We're stuck in a time loop.
Speed 2, I believe, was on a boat.
All right, video, go ahead and push in there. It's good.
Good picture right there.
Let me know what part you want to look at there.
This is perfect.
Perfect will have to do.
Yeah, that's fair. Perfect will have to do.
All right, sorry to inform you, but we're gonna take a piece of this just to
dang.
confirm. I know how disappointed you are. So
Worst
That's why
news.
she still has the
All right.
manipulator in her lap.
I actually just said that so you would get into a good position pretty close to the colony, so that if I decided, yeah, it wouldn't be too much of a trial. Yeah, so let's go and zoom out and see where we want to pick a piece out of.
Sure thing.
What was the yellow guy on the end?
Yeah, do you think you can lop off that crinoid with some branches?
Sure. Do you usually want part of that horizontal main base as well, or do you just want the vertical?
No, just the
pieces coming
vertical
off?
pieces. Yeah, yeah.
Okay.
If you grab, 4 or 5, pieces in your coral cutters snip, these are fairly heavy, so they're not going to be floaty,
Fairly
and
heavy.
it should sever pretty easily in the coral cutter.
Might be
Okay.
touching that.
Any thoughts on where it's going?
Let's go in the A compartment.
Yeah. A just has a rock in the bottom too.
Cool.
Where did the last one go? In B?
B, yeah.
B.
Yeah.
Yeah.
Okay. Yeah. If you can get it in there, let's do that.
Cool.
What type of coral did you say this was again?
So this is in the genus Calyptrophora, I think.
Okay.
Yeah, I think I spelled it in the chat. Nope. Let me write it down.
Just come back, make sure I'm all the way back.
The problem is that in Calyptrophora, usually the polyps are, when they retract and point up colony, or it's called polyp polarization. And they're all pointed up towards the branch tips when they retract. But in this case,
Video,
they're
you wanna
kind of
push in there a bit,
confused.
please?
There's polyps pointing all over the
That's good.
place. And we can get a crinoid with this one too, which makes crinoid people happy. Or people who study crinoids.
Crinoid people, I was like, is that a new,
New
is that a new
species.
genus? Yeah. Crinoid people?
New form of people.
Yeah.
Good idea for a sci-fi book.
Man, these
That'd be
they're
cool. Everyone flies around like brittle
Like
stars.
troughing it or something right now.
Or feather
Rolling
stars.
around.
Hopefully he's on this one.
It could be the brittle star versus the crinoid.
So you can see that when we touch the colony, the polyps will start to retract in one direction or the other. And I think the consensus is they all want to go towards the branch tips, which would confirm that this is in the genus Calyptrophora. Looks great.
You want to come a little wide there, video? Make sure that brittle star stays on there, or the feather star.
So I'm getting the feeling that this might be too big to go into A, so do you want to put it in the outer box? That yeah, so what's that number?
Starboard F.
Yeah, F.
Starboard F, roger.
Mm-hmm.
You guys want to zoom on this real quick?
Yes, please.
All right, video, go ahead and push in there a bit. All right, hold that. All right. You want to go a little bit tighter in there? Ooh. That's good.
So recently in this genus, the rationale for this collection is that in this genus, several just from the capstone collections over the past 3 years or so, several new species were described, and they all have slightly different branching
Science,
patterns.
is that good for you?
Yep.
All right, come
Yeah,
wide,
I
please,
think,
video.
we got captures. Yeah.
Mhm.
Okay.
Lots.
And so, so it would seem prudent that the more we collect, potentially the more species that we could describe. They seem to be extremely
I'm gonna do the
diverse in
same
this
land
area.
again. Sorry, Steve.
Sorry, did I speak over someone?
No, sorry. I spoke over you for a second.
Mhm.
I'm just gonna land real quick.
Yeah. And then, so after this collection, we're just going to make tracks, then we can track a line. How about that?
Yeah.
Okay.
Did I spell this one right? Calyptrophora?
You're on a roll today.
Man. Nailed it.
Footprint number 2.
Yeah, coming down.
Steve, can you tell us a little bit about what happens to these samples after they come on board and then where they go once we hit land?
Yeah, absolutely. So I'll talk about the biosamples and the geosamples a little bit. They're
Next to the
slightly
other
different
one.
in how we treat them.
Should I do it on top of it? Actually, let's do them step by step, yeah?
Yeah, it's a total side by side.
So you can do it nice and parallel.
So the biology samples, we take them up and we usually process them first because they contain pretty sensitive DNA that we don't want to expose to warm temperatures or dry air. So we typically will image them. The general flow is this: we'll image them. We will subsample them if they have associates. You know, in this case, the crinoid is an associate of the coral. We will put some pieces in various different preservations depending on what was requested, or but the general scheme is that we'll put everything in 95% ethanol, which is good at preserving DNA. So DNA can be extracted later, sequenced, and we can
Alright,
understand
you can go
something
ahead and
about their genetic makeup.
Go on, this guy here.
In this case, when the
What'd I do?
samples
I pressed the wrong
are
button,
processed, they'll be
No,
put
you're good.
into preservation and storage, and then when the ship returns to U.S. waters, they'll be shipped to the the mainland U.S. Or Hawaii, whichever one it is. They'll be shipped to the Museum of Comparative Zoology at Harvard, where all of the Nautilus samples
go ahead
are
and
being
bump her out.
deposited for
Bumping out.
request by the scientific community. So they are open access for those who are.
You said you wanted
could
F
be,
there?
Yes, please.
Yeah,
A little
forward.
vortex for my thrusters there.
Vortex.
You can keep it there, just so we can keep the other corals in their containers.
Sorry, which one are we going in?
We're going to F.
Roger.
F as in
Have
Frank.
a nice day.
So Steve, when a rock inevitably gets put on top of these, does that mess you up?
Yeah, we're gonna put a big So F I'm reserving for rocks. F for rocks, and E is gonna be for.
So you want this one in E then, not F?
I think there's already a coral in E.
There's
Yeah,
one
there's already coral in
E is the forward one, right?
Yeah.
Yeah.
that's the one I want.
Oh, okay.
Yeah, we can double those up. They're really different. You can't
Oh.
possibly confuse them.
What about any associates or anything?
There were no associates on the other
one?
Pleurogorgia, right? Yeah. In this case, we only found that the crinoid was on the primnoid, so we can separate that out.
It's a
Yeah.
bold move
Desperate
there,
times
Steve.
call for desperate measures. I want to leave one full box for rocks, a bigger box, just in case that's what they have to grab onto.
Good thing we got that pesky coral out of F
Yeah.
then.
I remember my first and second seasons here, Rennie, you probably remember too, the old bio boxes
Yeah.
used to have much smaller capacity,
Yeah.
and those things used to come up with like mixed sponges and debris. And I remember one time we sampled a coconut husk off the seafloor, and it just obliterated. It got coconut husk and wood debris everywhere, and we were trying to clean off the debris from the corals, and it was just a mess. So these boxes are significantly nicer for
They also
storing.
keep they keep them cold.
Yeah.
They're insulated a bit. Once it's shut, when you get to the top, they should be pretty cold in there. Still.
Yeah.
Well,
Yeah.
you guys would know, reaching your hands in if it's colder. The surface temperature of the water is like 29 C.
Yeah.
What are
Just
they?
crazy.
Forward box is very good at holding temperature, or it's definitely colder. Starboard box, not so much, from my experience. But I don't know if I don't know what that could be from.
What are they made out of?
You would know better than us, I think. I don't really know.
I have no idea. Some white stuff.
Yeah.
Sample number is 048.
Delrin, maybe.
048.
They don't look like Delrin.
They don't feel like Delrin. HDP? I don't know.
Yeah. Some high density super razzmatazz subsea.
Super razzmatazz.
Thank you very much. Very good collections.
Cool. Yeah. I think next one, you want to do it, Dan, so that I don't steal all this stuff?
What's that?
If you want to do the next one, I will share.
No. I'm happy to watch you operate that thing.
Cool. Okay, never mind. Take that back.
Unless we're doing one on the fly, then
Fly
I was
one
just
different.
teasing about the stick hog thing, because I was like that for decades.
Roger.
What's
At least
the
10
distance
years
now
before
to
you
waypoint 4?
still never, ever gets old.
500 meters.
Can you zoom out so we can see the platform? Waypoint 5.
Ah,
You're
sorry.
right above me on the button.
That's going to be a heck of a climb.
It looks like this is kind of a saddle, so they won't have to. They won't drop down too much, and then
Mhm.
they'll be able to go up. Perhaps.
And the distance between 4 and 5 is, or you know what? Not even 5. Just like what the shallowest point is, where that cliff gets flat again.
Here.
Might as well come up.
This way,
Gus,
Yeah,
and
directly upslope or
get the
whichever.
rest. Maybe you can distract Steve with something he hasn't seen yet.
Sorry,
Just
sorry.
to your
I'm on waypoint 4 here. And
Yeah.
you want
What's
to
4 to 5?
4 to
Let's have a
5.
look, yeah.
Okay.
There's a little bit
1300
left. You got to go
meters.
that
Okay.
way anyways to turn
Alright.
around.
Let's make tracks to 4.
Okay. I'm gonna go 0.2 knots. And we're
Point 2, you're crazy.
It's wild. It's gonna be slow at first, and then it'll steep. The slope will steepen, and maybe
I like 0.2. Anyone else? Second?
Yeah, 0.2 sounds
anyone
good.
else for 0.2?
Jess is going to show you the rest of the rock on the way out.
Great. This is just a really nice community. Can we get some like
It is
good
beautiful.
captures of that?
Could we track a line bearing 333, speed 0.2 knots?
See,
Thank
which way did Argus turn? He turned clockwise, didn't he? It turned clockwise.
Yeah.
Yes. Why are vehicles neutrally gender neutral? They're not a he, they're not a she, they're a it.
It's true. Although I keep on going between female and male for Herc sometimes. Depends
Well,
on.
if you look on the, if you Google remotely operated vehicle names, there's a little blurb in there why they're, or that they are gender neutral.
Huh.
Huh.
But ships are usually
Female.
Ships are female, yeah.
Because women can be allowed on. We kind of break that rule a lot.
All right, we are starting to track that line shortly.
So you said 333?
333.
Okay.
At a snail's pace.
I like snails. Snail's
Yeah.
pace is good.
So it was a peanut worm? Is that what you were saying?
Let me
Pop
double
down,
check
have a look, and then
that for
we'll
you.
get set up to go to 333.
Yeah, you got time.
It's kind of neat how they all
Some punculin. It's a punculin.
live under this rock.
Yeah, peanut worm, I think.
Peanut worm. So what do they do? Do they look like that their whole life?
Yep.
Yeah.
But you know,
Not entirely sure how they eat or anything. Kind of looking at that thing, wriggling around.
deep sea sipunculin.
You could probably start moving the ship anytime.
Oh
I
yeah,
think
we're
it's
moving.
underway.
You're moving?
Yeah.
Sweet.
We're, well, I called it in. We're not quite moving yet.
Yeah,
I missed that, sorry.
Pretty sure
No
it's not a
worries.
preep, that it's a sipunculin now.
I believe sipunculin comes from the Latin for small tube.
Huh.
I didn't know you spoke Latin.
Mm-hmm.
All right.
Cantate Domino Canticu Novum. Sing to the Lord a new jubilant song.
All right, kids.
Okay,
333.
Romani.
333.
333. That's the rumor.
Yeah.
Hunter,
Turn and burn.
turn and burn.
Start coming around too. Chase you around.
Yeah, it is like a mini road trip.
Was that you, video?
What?
The HD camera you
Flickered.
flickered on
flickered.
Herc?
I did not make it flicker.
You're gonna check the diagnostic sheet, which is this one here. Yeah, it's that little tracking pad there. Yeah, then it's the green box there. What are we looking at? 14, 13. It's weird. Yeah.
Did Herc's screen flicker for you guys back there?
I did not see a flicker.
They didn't
Nope.
see it.
That must have been our monitor then.
That's better than it being anything else.
Those are
Yeah.
nominal numbers, are they, Jess?
Yeah, 13 and 14 are good.
How do I get back to the this guy? No,
It's that guy there.
that dude.
Yeah, that one.
Oh, thank you.
All right, you want to start coming about on your head in there?
Coming about.
Thank you.
Yeah, thank you.
Want two?
I'll
Since
take
you
the whole bag.
You only got 2 samples, so you get a bonus.
Yeah.
1 for Rennie, 2 for you.
That's I get a prize. I get a prize just for being here. A for effort.
Me too.
I held the ship steady.
That is true, and that's a big help.
I didn't even do that. I outsourced it to the bridge.
It's fun driving the ship from the control room.
All right, so it looks like 333.
Yeah, I gotta. Pretty close.
If you want to come up a little bit as I come under you.
Sure. Spinny spin.
Roger. And driving forward now.
I'm less nervous about coming under at 20 meters because we've gotten away with it so many times now. As soon as I say that, bad things will happen.
Yeah, let's knock on some wood here. Thank
Touch
you.
wood.
That's the sand pattern there is pretty cool on Argus U. Would you mind giving me a porch on bubble when you get a
Sorry, yeah. Lagging. Well, that was strange. Another time shift.
Camera anomalies.
Getting close to the date line. Perhaps it's getting a little wonky.
Yeah,
Temporal
believe
distortion.
it or not. So as we cross between Kingman and Palmyra and Jarvis, we actually go ahead and then go back.
Yeah.
Yeah,
There's a little
technically.
the date line juts out because I think Kiribati wants to be on New Zealand and Australia time or closer to it.
Yeah, my phone was freaking out. I had to tell it to stop trying to change the time.
Yeah, I did.
Huh.
I had to go to manual.
Yeah, when we were coming over from San Francisco to Hawaii, we went through 3 different time zones, and my phone was like, okay, we're here now. And I was like, no, we're not.
Yeah, we usually keep the ship time to be the last port that we left until we get to the next port, depending on a few factors, but it keeps things easy. But all of our timestamps and all our recorded data, everything like that is all in UTC, so we don't ever get confused about that.
I do. It's real convenient when your phone changes the time during the middle of the night and you're using it for an alarm clock.
Oh
That's
yeah,
true.
that's the worst.
And we go on 24-hour time, or military time, which is so much easier to say 1400 than 2 o'clock in the afternoon, or 2 o'clock. Which 2 o'clock?
But it doesn't have that musical kind of lyrics to it, you know. It's kind of hard to say 1400 in the afternoon.
This is the musician coming out of you again,
Yeah,
isn't
Yeah,
he's
that's all you get.
practicing.
it?
You gotta.
Because that's all there is.
Well, that could be part of the lyrics too.
Yeah, American Samoan time is actually behind
Yeah, Samoa
Hawaiian
Apia is
Hawaiian
ahead.
Standard Time.
It is by I think an hour.
Yeah, an hour.
And then the other side of the line is tomorrow
yeah.
around the same time.
Yeah, when we fly back, we have to fly American Samoa to Samoa Apia, New Zealand, and then back to the States. So we go forward in time and back in time all in a day.
20 hours.
Make
for
sure
me
you
to
charge
get
up
home.
that flux capacitor.
Even more for me.
Really? Where are you flying to?
Connecticut.
Oh yeah, 'cause you're hopping across.
Extra
the-
6 hours.
Totally.
Yay.
I'm going via Fiji. For some reason. You guys, you go through New Zealand then?
Video
Yeah.
hours down a little bit there, please, Emily. Yeah, thanks.
You go to
Um,
Houston too?
Yeah.
Houston.
Yeah, it's they're taking us American Samoa to
that's
Samoa
why.
Apia, Apia to New Zealand, New Zealand to Houston, Houston, and then Boston, or wherever you need to go home from Houston. But Houston's kind of like the
It's a hub.
main point for a lot of people.
It's a hub for the Air New Zealand.
Yeah.
Going through LAX,
Yeah, me too.
Fiji.
Fiji, LAX.
I think that's the way a lot of people on the West Coast are going home.
Yeah, I think most of the cool kids are going through Houston. I'm
Yeah.
not-
No, all of the cool kids.
Should be approaching the kind of the base soon.
Yeah. Thanks, Ernie.
Yep.
That was a nice little rock detour. Good choice.
Yeah.
Good choice.
2 in a row.
Fish off to the left, actually,
Fish to left.
just off screen.
You want to go see it?
Yes, please.
Maybe it'll be like the fish from last night and just not move.
Oh, I see where you're seeing it.
Oh,
Oh, wow.
That was a special case.
I think that fish was petrified.
pretty stoic.
Reminded me of the time where I rode in Nautilus Live when I was little, and Greg was about to poke a fish, and they went to go poke it, and it swam away. And I go, "What did that fish ever do to you?" And 7 years later, they don't let me live it
Alright.
down.
Go ahead and push in a little bit there, video. Ooh, that's good. So now he's on a speed chase. Speed
Yeah.
chase.
All right, that's okay.
Roger.
You know, don't want to get too far off the slope.
Good flying.
Go ahead and come wide.
Whoa.
Wrangling fish with an ROV.
New pastime. There's a movie right there or TV show.
Is that another fish for you there?
Oh yeah, what's that one?
You wanna go see that one too, Steve?
Oh, where are you looking here?
Bottom of the right.
Oh, yes, please. Yeah.
Okay.
Sitting on the sediment. We've got some time before.
We got a little bit of time.
Yeah,
Yeah.
this 0.2 knots is a luxurious pace. Can do whatever we want.
Those aren't nodules, are they? Or round dudes? Are they just rocks?
All right, video. Go ahead
Hard
and
to say.
What do you
go
think,
slow.
Jen?
They look like they are covered in
What are
manganese.
you?
Whoa.
Look at the hairdo on that guy.
It's a tripod fish.
Yeah. of some
Weirdo.
kind. Never
There's a
seen-
common name for.
That's so cool.
Yeah.
It's a tripod reindeer fish.
Tripod.
Should
call him
flip over
Rudolph.
and still have a tripod.
Bathyterois is the name of that one. P-T-E-R-O-I-S. It's a silent P.
Very interesting.
We did see one of these on, I think it was the first dive.
I want to do a video sin and one bump down.
Yes, please.
One more bump. Ah. Look at your face.
So these
I
guys
can't see his eyes.
are
I don't know if he has them.
always, they seem to be always oriented upwards or headwards into the current.
Can brighten the situation a little bit.
Thank you.
And what they do is if they sense some food coming into the current, they ambush the prey, snatch it up, and then drift back down to the bottom.
Weirdo.
So it's kind of like a passive feeding in some ways.
Huh.
They just wait for the food to be delivered to them.
All right, guys, probably another few seconds, and then we'll get back on the road.
Looks like there's some damage on the caudal peduncle area and on the side next to the lateral line, but I don't see any parasites, obvious parasites here.
All right. If you guys are happy with this, I'm going to come wide.
Great.
Go ahead and come wide there, Emily. What a weird friend.
Cool coloring.
Yeah, very interesting. Sorry. You want to come full wide on Herc for a second? I'm going to make myself all squared up again.
On Herc or Argus?
On Herc, please.
Okay.
Argus too, I guess. Are we still zoomed in on? That would explain it. Thank you.
I'll go full wide on that.
All right, now you can go a little bit in on Herc there. Yeah, that's good.
Yeah, fun fact of video fun fact. The reason why it's so bright in that little patch towards Hercules, because that's where the lights are hitting, and the sand is kind of acting like one of those sheets that you hold up to like move light around to make places brighter. So that's why it is a lot harder to make it darker, because the rocks are really dark too.
Yeah, it seems I never noticed a hot spot in front of Herc before.
Yeah.
It's actually kind of nice that the lights aren't dimmable here.
Is that a cup coral or an anemone?
From an operator standpoint.
Did you want to see it, Steve?
Yeah, just like a quick zoom. You don't need to
Sure.
go in. I think it's an anemone. Anemone. Yeah.
Anemone.
All right, video, you want to
See
push
that
in there a bit,
there.
please?
All right, push in.
That's
Yeah, I'm gonna bring them that.
Looks like an
Anemone
anemone.
and sea-pen. Yeah. Thank you.
all right. Is that good for you, Steve?
Yeah.
All right, come wide, video, please.
Come on wide.
Coming up.
Anemone.
Just think of Finding Nemo.